This is the fertilization process leading to fraternal or identical twins. Now, when the sperm enters the female reproductive tract, it travels up to the fallopian tube searching for an egg to fertilize. This is the first start of potential conception. As sperms penetrate the outer layer of the egg, only one sperm can successfully break through this layer. And once it does so, the surface of the egg changes immediately to prevent any sperm from entering. When the sperm enters the egg, the tail is no longer needed and thus disintegrates. The head of the sperm fuses with the egg. The sperm's nucleus, which carries the father's DNA, fuses with the egg's nucleus, which contains the mother's DNA. The genetic material from both parents combine to create a complete set of 46 chromosomes, half of which are inherited from each parent. This single cell is known as a zygote, which is the first cell of the developing embryo. It carries all the genetic instructions necessary to form a new individual. The zygote begins a rapid process of cell division. It divides into 2 cells, then 4 cells, then finally 8 cells, forming a small growing balls of cells. As the cells are dividing, gentle muscle contraction in the tube walls, tiny hair-like structures called cilia, which propels the zygote along the fallopian tube towards the uterus. This journey typically takes about 3 to 5 days. By the time the zygote reaches about 16 to 32 cells, it becomes what we call the marilla, which looks like a tiny cluster of cells. By 4 days of development, the marilla begins to absorb fluids and a large cavity called the blastocyte forming within it. The blastocytes has an outer layer of cells that will eventually form the placenta, and the inner group of the cell that will develop into the baby. When the blastocyst finally reaches the uterus, by day 5, it hatches out of its outer membrane. The blastocyst attaches itself to the uterine lining known as the endometrium walls. This process is thus called implantation. This allows the blastocyte to attach to the uterine wall, enabling it to obtain nutrients and oxygen from the mother's blood supply, supporting its continued growth and development. In rare and mild cases, unusual events can occur during the early stages of cell division. The developing cells split into 2 distinct groups, each with the same complete sets of the DNA from the original fertilized egg. These 2 groups of cells continue to divide independently, and each one will eventually develop into a baby. The exact mechanism that triggers the splitting of the zygote is not yet fully understood. Potential genetic factors, along which contain environmental influences such as the specific fertility treatment, may affect the likelihood of disappearances. Identical twins can form at any point from the first couple of days after fertilization up to about 2 weeks. The split timing determines how much the twins share within the womb. The earliest separation is believed to occur at the 2-cell stage, in which the separate zygote develops. This early split leads to each twin developing separately with its own amniotic sac and placenta. These are called the dichotomic amniotic twins. Each twin has its own environmental within the womb. This is the most common and lowest risk scenario for identical twins. If the split occurs a bit later of 4 to 8 days after fertilization, usually after the morilla has developed into a blastocyst. These are thus called the monochorionic diamniotic twins, meaning they share one placenta but each has its own amniotic sac. This setup occurs in most identical twins pregnancies and results in the twin leading to a shared blood supply but separate pockets in the uterus, showing a placenta can result in complications like twin-twin transfusion syndrome, the triple TS. In this condition, one twin receives most of the blood flow and flow to the others, and thus flow is compromised. As a result, one twin is larger than the other. The outcome is poor, with each resulting to death of birth twins occurring in 50 to 70% of cases. However, because of each twin has its own amniotic sac, they are protected from becoming entangled with each other. The umbilical cord, which reduces some of the associated risks. If the split occurs after the blastocyst has fully implanted in the uterus, 8 to 13 days after fertilization, it is thus known as the monochorionic monoamniotic twins. This arrangement is more complex because the twins share not only placenta but also blood supply and thus high risk of triple TS. But they also share amniotic sac. This share amniotic sac increases the risk of umbilical cord entanglement as the cord have more opportunities to cross paths. In extremely rare cases, the split occur even later between 13 to 15 days, resulting in twins who are conjoined because they did not entirely separate. Conjoined twins split too later for each to develop entirely separate bodies and share some organs or body parts. They face significant challenges due to their unique conditions. Most of them are stillborn. Of those who survive, around 35% only live a few days after birth due to complex health issues. Those who survive often faces complex health and modified challenges. These slight variations in the timing of the split influences each twin's prenatal environment, but the essential process remains the same. One fertilized egg or zygote splits, resulting in two genetically identical babies. Wow, this is Dr. Eddie Ongano, School of Health. If you find this video interesting, please like, share, comment, and remember to follow. Thank you. Let's meet in the next video.